Dr. Larry Smarr is another very, very, very famous guy. He's done a lot of different things in his life, ranging from general relativity to what he's doing now, which is running the CALIT2 Institute, which is probably the most amazing institute in the country. Larry, would you like to come out? How are you?
Great.
Have a seat.
Sure.
Larry is known for many, many, many things. I won't list them all because it'd take all day. One of which is you're known as being the most measured man, if that's the right term.
The thing that I'm most proud of is that Lee Hood refers to me as his lab rat.
Duly put, well put. So Larry has measured, you've been measuring your body for 10 years, more?
Intensively for 10 years.
Yeah, okay. On a very regular basis, lots of parameters. Mm-hmm. And so literally Larry's body is the most well-documented from a scientific perspective, shall we say. I've actually handled a plastic model of Larry's colon, but we're not going to talk about that today. But we've all had our own experience learning about Larry's body. He's been very generous to share with us and with the world, actually. And so the data sitting in this chair right here over 10 years is invaluable. And Larry, among all folks, has taken this much further by studying it in many, many different ways. So not only does he share that information, but he studies it. Would you like to talk to us a little bit about that work?
Yeah, I mean, I spent my youth as doing relativistic astrophysics, which is integrated dynamic systems like Lee was talking about. And I just assumed that that's the way we'd think about humans because they're actually way more complex dynamic systems than what you see up in the sky. But instead, we've broken them apart into pieces, and it's the putting them back into a system and understanding the deep amount of data that is now possible to be generated over time. In my case, say 100 and something variables in my blood and stool, but also to understand the microbiome by looking at literally billions to trillions of numbers per person over time.
Meaning the gut bacteria that we all carry.
Yep.
Which is 90% of the cells in our body.
Of the DNA cells,
yeah.
Yes.
Yeah.
Mm-hmm. And so, we think we know who we are.
Clicker, by the way, if you want to use.
But we really don't.
Yeah. This is new science.
It is.
Yeah. And exciting.
So, let me show you a little bit about an example of how we use very large amounts of data and then work with pattern computer to find new things that we didn't really understand before about the difference in health and disease. Given that 90% of the cells in your body with DNA are not human, they're microbes, and they're not a part of medicine today, bringing these into medicine as the exponential to decrease the cost of sequencing and to decrease the cost of computing continues into medicine is going to totally disrupt it. So I'm just going to show you one example. I've been doing many different kinds of things, but this is just one experiment over the last 5 years where we downloaded very deep genetic sequencing of 250 subjects, each different people. Myself, 7 time points, I had found out by doing these kind of things that Lee was talking about that I had colonic Crohn's, a form of inflammatory bowel disease and autoimmune disease. And then we downloaded a number of examples of the other 2 kinds of IBD, ulcerative colitis and ileal Crohn's. That was about 2.7 trillion bases of DNA. We then put that into a very elaborate software system that Weizong Li, my collaborator, developed. We ran about 200,000 CPU hours on the San Diego supercomputer, which is about the same as running your computer 24/7 for 25 years straight. With that, you could convert the raw reads that come out of an Illumina sequencer of, say, in this case, stool samples that 40% but dry weight is actually microbes. And so you're sequencing their DNA. You rebuild from that what the taxonomy, that is, you are in ecology, a very complex ecology. Like you look at a forest, it's oaks and maples and poison ivy. Okay, so how many of each of those are there? That's taxonomy. But each of those has genes, and the average microbe has, say, 3,000, 5,000 genes. So there's actually a couple of million genes in the microbiome compared to, say, 20 some odd thousand in your human genome. So most of the ability to convert from a gene to a protein, which is the business end of what goes on in the biology, is really unknown. Now here's an example. I've taken just about 32 of those 250 healthy people on the left, and then the ileal Crohn's, ulcerative colitis, and the colonic Crohn's on the right. And those are stacked bar graphs of the composition of the gut microbiome, where you have about 30 or 40 trillion microbes in your large intestine. And the blue and the red are two so-called phyla. So like phyla, if you were thinking of macro animals like tigers and jellyfish and insects are like different phyla. So these are very wildly different bacteria. But the red and the blue are sort of 90%. Whereas in the disease state, you notice the blue is completely gone, 50-fold reduced of the most important phyla. That's one reason you're sick. And the ulcerative colitis is proteobacteria, E. coli, the light blue that shows up. And then in me, you see over time, the blue is pretty well suppressed except for that one time in 6. I'll blow that up a little bit. It's healthy again on the left, and the blue and the red, you see the blue dominates. And then that's 1, 2, 3, 4, 5, 6, 7 times over a year and a half of my stool samples that have been genetically sequenced. And what you can see is, where's the blue? And it pops back up on just one time and then goes back again. And this dynamic episodic behavior that we know is associated with IBD, but then now you can begin to read it out in terms of the actual evolution of the microbes. Remember that 6th point. Now if I look at it in terms of a clustering diagram, this is called a principal component analysis. This is work that was published a year and a half ago. Then you see the different colored dots are the healthy and the 3 different disease states, and they completely separate from one another just based on not the taxonomy, but now we've gone to the actual genes. Notice that this is called KEGG, K-E-G-G. That was in Lee's slide earlier. These are protein families, 10,000 protein families across the microbiome. And when you then do the analysis on those, you separate things out. So I gave this data set then to Pattern Computer. And over the last few months, I've worked with their technical team to say, if you had this data, what would you discover that I haven't seen? And the first thing they did is, by using the Pattern Computer, they were able to take this 10,000 variables per each of my 60 people and say, well, there's 39 out of those 10,000 that actually preserve most of the structure. That's an amazing reduction, almost 3 orders of magnitude. Then they were able to correlate those using the classic Spearman cross-correlation analysis, and you can see the 4 blocks down through the diagonal are at lower right, the healthy people, and then upward through the 3 disease states. You'll notice those 2 horizontal yellow bars where it seems like there's something different about them. And so when we go back and look now at a 3-dimensional version of that principal component analysis, that clustering diagram, you can see that on the left there that the blue, the ulcerative colitis, completely separates away from the red ones, which are the colonic Crohn's, that's me, and then the ileal Crohn's, and then the healthy are all over one side. But what is that group in the middle? In other words, that sort of seems like those are patients who might be in a region that's close to disease, just like we saw with Ben Brown, where those 2 people, one was close to being healthy and the other was quite deep into it. Well, that's the same thing you're seeing here. And so what the Pattern Computer people came back and said, Larry, we need to look at this yellow bridge set of samples, which didn't occur to me before to think about that. And when you look at them, you can see, once again, this is now showing the direction, the arrows are connecting the 7 dots of me, and you can see that literally that number 6, just remember that one, it did swing away from the essentially disease side to where the colored dots are to the left into the healthy space and then back again. And this is, again, the way your body's trying to get back to health, but then the underlying thing is carrying it back into the disease state. So we said, okay, let's take these 39 of the 10,000 KEGG protein families that the pattern computer identified as important, and say which of those show a gradient across that yellow space. So these are which of those 39 protein families seem to have a directional change from disease to health. And here you just see one of them, and that's the gradient on the left. And you can just see it's a beautiful pattern that was completely unknown to me. And then on the left, you can see the colors are going essentially in the color of the rainbow from one side to the other, from the healthy side to the disease side. And here's two more. And again, you're finding these that show this gradient across there. So we've now selected just nine out of 10,000, so a 1,000-fold smaller number of variables to look at. Many of these KEGG protein families are not known what they do. By the way, KEGG is the Kyoto Encyclopedia of Genes and Genomes. So they are a group that have very carefully annotated everything in the literature that mentions these particular proteins. And what we find is that these that were selected by the pattern computer were discovered are associated with oxidative phosphorylation, which is a disease, actually, if you don't have enough of those proteins, and is involved in a variety of the things that are associated with disease. So summing it up, basically, there was a 1,000-fold reduction in the scale of the problem. These 9, we are now going to be able to go off. There's a hypothesis these 9 might be important. So we are now going to go back and start doing work on discovering better what those proteins do. This particular hypothesis has been emerging that this abnormal oxidative phosphorylation, or oxidative stress, you may have heard more about, is an important part of the disease. And of course, there's literature on that as well. So let me tie this back now to Lee. You remember he was talking about these networks. And this goes back to what Ben was saying about the parts list and then the assembled car. We've had 100 years of reductive science. And that's been very helpful. We found out there was DNA. We then found out there was the three-dimensional structure of a double helix. We then found out there were genes along it. All that's great, but you've got to put it back together into the integrated dynamic entity that you broke apart in the first place if you really want to understand why is there a healthy state and why does that transition into a disease state and what can I see early. This is work that inspired me. I've worked with Lee for over 25 years. When he did this work, which was published in 2009, but he was already doing it back when I first started on this, this was introducing prions into mice and then tracking these. Each of those little yellows is one of these protein families we just talked about, these KEGG things. And notice that they're arranged in an interactive feedback network. And what you see is time going across. And once you perturb one of those, it then perturbs the next one and the next one. It's like a pachinko machine until finally you get all of those red dots, and that's when you finally notice you have a symptom. But what was going on was happening and was measurable all that time before, and you could have gone in and put one of these things back and you would have been fine. That is why all your cars work. You keep longitudinal time series information on each of the vital components of your car. You compare it against the population at regular intervals, and when you see something starting to go out of whack, you make some change, it goes back, and now your car at 200,000 miles is healthy. That's where we're going with humans with healthy longevity.
Thank you. Perfectly done, as usual. So I'm so proud to be a friend of yours and to know what you're doing.
Well, thank you. And one of the things I'd like to say about Mark is in his role as creating the strategic news service, he actually gave me the opportunity to publish the first article on quantifying my body. because I couldn't get a journal or anything to pay attention to it. And after that, publication of that, everything else that I've done flowed.
Yeah, well, I'm happy to have had that happen. So to summarize what I think you've just told us all of this work you're describing, and I think you described our role in it quite well. What's your experience in working with the team, and how do you see it going forward?
Well, of course, as part of my job, I have been working for 30 years with all the vendors and computing and so forth. This was one of the most enjoyable collaborations that I think I've had in many years. The technical team in Pattern Computer worked with me for the last couple of months on a very tightly iterative thing. They would find things. I would say, what do you think of this, Larry? And so as a domain expert working with the pattern discovery experts, we were able to iterate against each other back and forth, back and forth, back and forth, had weekly phone calls for months. And in fact, there's a write-up of what I just talked about on the Pattern Computer website if you'd like to see more about it. So it was a very, very good initial engagement, but we're going to need this a lot more, even as just this year goes by, because I showed you basically about 60 patients, and we were looking at about 10,000 protein families. So imagine imagine your medical record with 10,000 entries for each person that are these different protein families. But by the end of the year, we're now sequencing the stool of what will be about 500 instead of 50 or 60 people, so 10 times more patients. And instead of just the 10,000 protein families, we're going to be having anywhere from 1 to 2 million relative abundance of the actual genes themselves, not grouped together into these protein families. So about 100 times more variables per person, 10 times more people. So that's 1,000 times larger data set in this calendar year that we've got to analyze than what we just went through, which was, if you think about how would you as a human look at 10,000 variables across 60 people and see the pattern? All right, now imagine 1,000 times that, and that's what this is this year. So that's the whole point. This exponential is not stopping.
Right.
And the ability to look at the system integration is and make discoveries that way is really, that's what the future is about.
Key. It's key. Yeah. And I wanted to underline one thing that you said, because I think it was critical for other scientists, and that is, did this collaboration help you to refine or change your pathway, your hypothesis, how you saw the work?
Right. Well, the very fact that they were able to come up with a specific biochemical pathway involving this oxidative phosphorylation, that now we're going, that was not on my agenda.
Yeah.
Going forward now, we're going to look at that quite deeply and how it plays out in people. And of course, now you can do lots of lab experiments as well.
Yeah. Thank you, Larry.
Well, thank you.
So